So this is DNA replication is one of those topics that one of the many topics that when I started teaching biology that I hated to teach because it didn't seem interesting to me. Believe it or not, college does not teach you everything you need to know about the subject. Okay, most of the stuff that I've learned has been just through teaching, because no one wants to stand in front of people and not exactly know what's going on. So DNA replication. I'm going to expand this picture right here, and I'll bring it back so you guys can check it out. All right. So, do you recognize the cell division, the cell cycle?
Yes.
Okay. So, from here all the way around, this is interphase. Okay. And then interphase has G1 phase. That's where the cell's growing and it's doing whatever the cell thing it's supposed to do. Synthesis phase. That's where it's actually replicating its DNA or duplicating its DNA, and then the rest phase, and then it goes through mitosis. So interphase, prophase, metaphase, anaphase, telophase. So if you understand all the cell cycle, then you'll understand, S phase, interphase. This is where all of that takes place. Let me take this back down for you. Okay, so make sure you know, and I can't remember if this is a test question, but it happens during S phase of mitosis, and then it's where one DNA strand is duplicated into two complete. Now think about it. You start out with a parent cell, and it's at that point where it's getting too big, right? Cells can't be big. And so it has 46 human chromosomes if we're talking about a human cell. And during the S phase of interphase, it goes from 46 to 92 chromosomes. So it's duplicated it. That way when it divides, each cell has 46. And it's all because of this. I had an AP student ask me, it was a great question. He said, what would happen if replication stopped? I said, well, think about it. If you got cut, you wouldn't heal anymore. You would stop growing. So if it stopped right now, you guys would never get even a millimeter taller. And your skin cells, they're always coming off. Well, if they keep coming off and there's nothing to replace them, eventually you wouldn't have any skin left. And so this has got to keep going. Now DNA replication, if I were you, I would know in my mind that it takes 8 hours. That's a rough number, 8 hours. Okay? So going from 46 chromosomes to 92 using DNA replication, 8 hours. Remember, the whole cell cycle is like 20, 21 hours. But 8 hours of it spent just duplicating DNA. Okay, any questions so far? Okay, so there's a picture. Now, I'm sure in college they told me this, but I didn't pay attention, that when a chromosome replicates, when your DNA, it doesn't start at the very beginning of the DNA strand and go in a line to the end. It doesn't do it. If it did, it would take 6 months to go from one end to the next. So if it did take that long, you'd cut yourself, you wouldn't heal for a long time. And you would grow very slowly. And your skin cells would not replace very quickly. So Mother Nature has found a way to do this in 8 hours. So instead of 6 months, 8 hours. Here's how it does it. Okay, these are called replication bubbles. Write it down, replication bubbles. This is not general biology stuff. When I first started teaching biology, I would never have taught this, but I think you guys can understand it. So instead of starting at this as the DNA strand, instead of starting at this end and flying that way, it may start in the middle. And then another one will open up somewhere else, and another replication, and you can see what's happening. They're going opposite directions. Okay, so it's going to start going this way. So if you're talking DNA strand, I wouldn't think it would be out of the realm of possibility. You would have 1 million replication bubbles zooming through, creating 2 new DNA strands. Now what they've done here is they kept dotted lines here. This is the new strand. This is the old strand. Okay, Mother Nature always keeps 2 of them in reserve. So this one is in reserve, this one is in reserve, this is the new strand. So you start with this, you end up with this. So replication bubbles. Now, you see the dots? If I were to draw a line between, I guess, that one and that one, those are the base pairs, right? So adenine, thymine, or cytosine, guanine. So this travels, DNA replication takes place at 2,000 base pairs per second. Think about that. That's fast. That is extremely quick. Okay? And it does make some mistakes. Now, if I put on my jacket, I don't know. Really, what do you think? Is there a lot of like little teeth on this zipper? Wouldn't you say there's probably 1,000?
Sure.
And if we're talking 2,000 a second, and I'm doing 1,000, there's 1 second, and I've got 1,000. Can you imagine doing? And that's happening constantly. Here's a cool thing. Even though it happens so quick, there is one mistake in every 1,000 base pairs. That's pretty good. Would you take that? I wouldn't, because you're talking the whole human genome is 3 billion base pairs. If you get one mistake every 1,000, you're going to have 1,000,000 mistakes. Okay? But Mother Nature's come up with an answer to that problem too. And I'll tell you here in a second. Your body's doing a really good job of it. Mine not so well. Now, 2 players in this. Remember when we talked about at the very beginning of the year when we were talking about carbs and nucleic acids and lipids and proteins? I said if the word ends in biology with an OSE, what am I talking about? Sugar. But if I say ASE, what am I talking about? Enzyme. Okay, OSE is sugar, like glucose, fructose, sucrose. And then there's ASE, those are enzymes. Think of an enzyme as just like a protein machine. It's a machine made of proteins that does one certain function. Okay, it functions at the right temperature, the right pH, whatever. Well, if I were to ask you to describe the shape of the DNA molecule, and I said double helix, right? And everybody knows that the helix is a coil. We talked about it the other day. Well, look at the name. Helicase. Helicase, helicase, however you want to pronounce it. Its purpose is to break the bonds between the adenine and the thymine, the guanine, the cytosine. And what it's in effect doing is it's taking that coil out. It's uncoiling it as it breaks it open. Okay? And it's doing it 2,000 base pairs every second. That's pretty cool. And so when come test time, helicase, double helix. Oh, enzyme that breaks the coil and opens it up. Remember, when you get an adenine near a thymine, they click together like a magnet. Think of it like that. It's not exactly that way, but it's good enough for us. But what would happen if I got an adenine near a guanine? Have you ever tried to put 2 same sides of the magnet together? Depending on the strength of the magnet, you're never going to do it. Okay, and that's what's happening here. This going through, breaking that little magnetic bond. Okay, here's the other one that you need to know. There's more. There is exonuclease, there's ligase, there's primase. There's quite a few more that you'll learn when you get to AP Bio if you go that way. But these are the 2 main ones. DNA polymerase. Well, you guys know that DNA, RNA, it's a polymer. Big, long chain molecule made of monomers. Well, DNA polymerase, it's making 2 long chains. So here's your original DNA helicase is zooming through at 2,000 base pairs a second. And then your polymerases, they're coming through and they're reading it. And they're just like, oh, that's a G. What goes with a C? Boom. A. Oh, that's a thymine. Thymine. Oh, A. So it's placing everything it needs. So it's reading, placing, and reading again. And then it moves on. 2,000 a second. Think about that. It reads, places, proofreads, and it still makes a mistake 1 of every 1,000 times. So it makes 1 mistake every second. Every second. And that's what its purpose is. So if it detects an A, an adenine, it's going to go find the corresponding base pair, the thymine, and it's going to place it in there. Okay, and then you have your 2, and remember, this is going to go to one daughter cell. This is going to go to the other daughter cell. And this is not, We're talking mitosis here. Growth, repair, replenish. But a key thing here, it also repairs mistakes. Here's a wild thing. There's more than one DNA polymerase. There's I want to say a 2 and a 3. I can't remember exactly. But what they do is they come through and they check and recheck. And you guys have written papers, right? Are you supposed to proofread it yourself? Is that the best? No, because you're used to your style of writing, right? And it's always nice to have somebody else proofread it, even though you do. So you wouldn't want to turn in your first draft. You'd want to get it proofread. Somebody else could see something different. And so this thing proofreads, it misses some every once in a while. And then it has somebody else, another one comes through. And that's just the way it works. Now, 3 types of errors that can happen here that you need to know. Sometimes errors take place in a very critical, let's say a lady is pregnant and just newly pregnant and the baby is forming really fast and this replication is going on inside the baby. If there are, maybe some kind of a chemical medicine in there, it might interfere with replication. And so you might get something like a skipped nucleotide. If it's not fixed, then at a critical time there might not be a protein formed and the baby might spontaneously abort. So most times when people miscarry, it's something along this line. There's something going on here, but there's a skipped nucleotide. If it's not caught, it becomes something really crazy. Okay, so there's a skipped one, wrong one. Okay, doesn't always happen that way. We talked about it, right? You put a thymine and adenine together, they click together. But if you have an adenine and you got a thymine and a thymine, there's going to be something off there that's going to be wrong, or it could be an adenine thymine cytosine, and you get that hump in the DNA, and then if the DNA polymerase leaves it, then another one comes behind. If it doesn't correct it, then it becomes something that eventually is going to lead to something bad. Okay, last one. Wrong one. Wrong one. So an adenine with a cytosine. That's what's going to happen. Okay, I'm going to turn the light on, and I'm going to turn it back off when we finish. If you look at me, my skin used to be smooth, no spots, no freckles, no anything. Right? My hair was dark black. You don't believe me? Go look at the picture when I was 30. And now look at it, it's gray. So all these mistakes that you're seeing here, my body is accumulating. It doesn't repair them. And the bad thing is that if it doesn't repair them, when my cell goes through replication, the mistake is duplicated. And so you keep getting these additions, and your hair will turn gray. It's happening to you right now. But right now, you don't have as many mistakes as I do, and I don't have as many mistakes as, say, like somebody who's 110. They've got lots of it. Okay? And so this is happening all the time. Now, if it's not repaired, it becomes what we're going to study next chapter is a mutation. Mutations, you have mutations in your cells right now. They're not necessarily bad, but they're going to start adding up, and then you will start showing them. Now, you see where I got that 3 billion number again? Think about it. I said DNA polymerase makes a mistake 1 in every 1,000. I can't remember the number. And you have whatever base pairs, 1 in every 1,000 base pairs there's a mistake. But look, that's 3 billion. Remember, that's how many base pairs are in the human genome. It is so good. Our system is so good that 1 in every 3 billion base pairs, there's a mistake. Think about that. In 8 hours time, zooming 2,000 base pairs a second, out of 3 billion times, there's only 1 mistake that's never repaired. That's why we age, I guess, rather slowly. Okay? And that's why that number's there. Again, we're going to talk about mutations here later. Now, I need everybody to come up here and grab one of these. Everybody needs one. They're the same thing.